All right. So to continue from our discussion about cell cycle, particularly continuing on from our discussion of interphase, we are going to start looking at the other part of the cell cycle, the last phase, which we generally refer to as mitosis. Mitosis is a specific type of cell division that occurs within cells. So, the important part of mitosis is the nucleus itself. We have to remember that the job of the nucleus is to contain the DNA of the cell or the organism. In an organism, every single cell, every cell needs to contain the full DNA of that creature. Every single cell, no matter what its job is, has to have the full DNA blueprint of the organism. So, even though if we think about the cells on our skin, even though they're skin cells, they still need the DNA that would make brain cells or muscle cells. Certain events happen that determine what it is, and we'll actually look at that more later. So, since every cell needs a full set of DNA, as we mentioned before, during interphase, particularly what we call S phase, the DNA, or the cell I should say, the cell makes a copy of its DNA before it divides. That way, both cells have a full copy. So it makes a copy of its DNA during interphase. That way, both cells, both daughter cells after division, will have a full set of DNA. So, to separate DNA, we can't just separate the molecules, because they're all really kind of interwound. The DNA molecules are extremely, extremely long. So what happens is they wind into these tight bundles. So I want you to imagine that you take a spring, right? And you start twisting it, and you twist it, and it kind of starts kinking over itself, twisting into these tighter and tighter and tighter packages. Same thing happens. And it creates what we know as a chromosome. Chromosomes are distinctive packages of DNA that have been tightly wound to prepare for cell division. And you can kind of think of chromosomes. If we think of the DNA of our cells as a blueprint for us, well, for extremely large and complex buildings, generally these blueprints have multiple pages. One page will detail the electrical systems, one will detail the plumbing, one will detail the structure, etc., etc. It's kind of the same way with DNA. The chromosomes are the distinctive packages of DNA. They're the distinctive pages of each blueprint. Okay, each one has directions to build something else. And all pieces need to be there because some pieces have information for other pieces. So chromosomes have several parts. They have each of these kind of stick-looking things is what is referred to as a chromatid. And there are two chromatids. We call these sisters because they are identical copies of that chromosome. So a chromosome is a set of the duplicated chromatids. And it's held together with what is called a centromere. And just because it's labeled on this diagram, the end caps of DNA, and these are kind of protective pieces, these are what are called telomeres. We don't really need to worry too much about them right now, but just so you're aware. So, for division to occur, these chromosomes must be tightly packed to ensure that there is an equal division of the DNA once the cell divides. So these are tightly wound and packaged. That way, when it's split, we can ensure that there's an equal split. That way, there's not chunks of DNA that are just missing. So, real quick review. I want to pose a question for you guys. Which structure connects sister chromatids together. And this is what gives the chromosomes their X shape. So what structure connects the chromatids together to give us that X shape? Okay. Excellent. Let's move on. So the phase that we are actually looking at, if we go back to our cell cycle, there are 4 phases of the cell cycle. Well, 2 phases, I should say. There's interphase, and then what is called M phase. M phase is what we are looking at right now. M phase is the actual phase that the division of the cell occurs in. Okay, so this is where the actual cell division occurs, is during M phase. The cell spends a significantly smaller chunk of time in M phase than it does in interphase. Because if you remember, interphase, not only is it when the cell is growing and duplicating and preparing, but interphase is also when the normal functions of that cell happen. So we want division to be as short, as quick as possible. That way, cells can go back to doing their normal thing. And there are 2 parts to 2 parts to M phase, I should say. There is mitosis. Mitosis is the division of the nucleus in the DNA. So, the division of the nucleus occurs in several phases. Okay? We're going to talk about 4 in particular. Prophase, metaphase, anaphase, and telophase. So this is the actual division of the nucleus. The steps that I'm going to detail, walk through what has to happen in each step. That way, the cells can then divide. So mitosis is the division of the DNA and the division of the nucleus, whereas our final step, part 2 of M phase, which is called cytokinesis, can occur. Cytokinesis is the division of the rest of the cell. It's the division of the cytoplasm, hence the name, the cell parts, so the other organelles in the cell, and eventually the cells as a whole. So, I want to go through each step of mitosis point by point. The first step, and this is in fact the longest step, is prophase. So many scientists actually split prophase into 2 parts because it is so long and so much happens in it. We're going to talk about it as a whole, but I'm going to detail what the 2 parts of it are. So this is a cell that is currently in prophase. Prophase, during this phase, the main thing that happens is that this is when the DNA condenses. This is when chromosomes become visible. It is no longer just a mass of DNA. It is now those actual tight bundles begin appearing. And you can see this in the cell slide directly below the diagram. You can kind of see how there's these gaps that are appearing within the nucleus. Those are the chromosomes forming. So the chromosomes form and the 2 sister chromatids, they attach at that centromere. The second main thing that occurs, and you kind of see these structures in the diagram. It's much harder to see these in the slides, but these things begin appearing. These are what are called centrioles. Centrioles are what put together something called spindle fibers. These are the actual, ropes. Imagine you're trying to pull something apart. If you have a rope tied to 2 sides, you can pull it apart. That's what the spindle fibers do. The spindle fibers form very, very early in mitosis. So these spindle fibers and the centrioles appear very early. Then the chromosomes condense. The centrioles align themselves so they're on opposite sides of the cell. And lastly, what we can see here, if you notice that the nucleus kind of has this dashed line around it, the membrane around the nucleus disappears. That membrane needs to disappear so that way the cell can divide. So that's step 1, prophase. Step 2 is called metaphase. And metaphase actually tends to be the shortest of the phases of mitosis. During this phase, what happens is that chromosomes align along the center of the cell. So they align right along the equator, you could think of it. They line up opposite sides, or they line up along the middle with the centrioles on opposite sides. Second thing that happens during this phase is, as you can see in the diagram, and you can actually see this happening in the cell below it, those ropes, those spindle fibers, attach to the centromeres of each chromosome. So they attach that center part of the X of each chromosome. Once this happens, the chromosomes are now ready to divide. And this is when one of the checkpoints of the cell cycle occurs. The cell checks to make sure are all the spindle fibers attached? Are all the chromosomes aligned? Okay, we're good. We're ready to go. And we move into step 3. Step 3 is called anaphase. This is our third phase of mitosis. This is when the actual separation occurs. Once metaphase ends and it's ready to go, boom. The spindle fibers pull, and they pull apart those chromosomes. Chromosomes are then pulled to the opposite sides of the cell, and you can actually see this really well in the cell down below. You can see those chromosomes being pulled apart. And then the spindle fibers themselves begin to break down. As they're being pulled in, the centrioles start to deconstruct them because they're not needed anymore. Once they pull the chromosomes to the opposite sides, it breaks everything down. This phase ends once the chromosomes reach the opposite sides of the cell. And we move on to the fourth and final phase of mitosis, which is called telophase. Telophase is when the nucleus begins to reform. Okay? So what happens is those chromosomes begin to unwind back into they begin to unwind back to normal, back into the strands of DNA, which, if you remember, is what we call chromatin. And that nuclear membrane, that membrane around the nucleus, begins to reform. At this point, the process of mitosis is complete. The separation of DNA and the division of the nucleus has happened. There are now 2 distinctive nuclei and 2 distinctive sets of DNA, one for each of the new daughter cells. And this is also when the cell begins to divide into 2 distinct cells. So, before we move on to the last part of M phase, I want you to, after us having finished those 4 phases, give me a brief, really brief, 1-sentence descriptions of what occurs in each of the 4 phases of mitosis. Give me a brief, 1-sentence description of what happens in all 4 phases of mitosis. Okay, so now let's move on to the final piece of cell division. Cytokinesis. We've separated the nucleus at this point. So the only thing left to do is divide the remainder of the cell. That's what cytokinesis is. Cytokinesis is the division of the remaining cell materials. This includes the cytoplasm, this includes nutrients, this includes organelles, whatever else needs to be divvied up. This actually begins to happen during telophase. So at the end-ish of telophase is actually when cytokinesis starts to happen. And I'm actually going to talk about 2 different sets because cytokinesis is different for animals as compared to plants. So what I have up here is a plant cell, and I will come back to this here in just a moment. So during cytokinesis, as we already said, the cytoplasm and the materials are split into 2 distinct daughter cells. For animals, animal cells are what I have in pink on the left. the membrane, there's kind of this structure that sort of winds around in between the two cells. So imagine like if you're trying to separate a balloon into two things, or like imagine a balloon animal. You take the long balloon and you spin it, and at that spin, it ends up separating it into two essentially distinctive balloons. That's what happens in animals for cytokinesis. There's this structure that kind of winds the cells up and up and up until the cell membranes connect, and then they separate. For plants, on the other hand, their membrane is not that flexible because the cell wall adds too much structure. So what ends up happening is a cell wall begins to appear, and that's what you can see in yellow on the right. That cell plate, we call it, begins to appear in between the two cells, and it forms into a full cell wall, and the rest of it is, well, the membrane kind of wraps around it. And you can see this in the diagram that I posted here as well. On the left side is our animal cell. We have this ring that kind of tightens and tightens and tightens and tightens, creating this furrow until they connect and they pop apart. On the right side, we can see this cell plate forming and joining with the membrane on each side, and then the membrane just kind of slowly drifts down, cutting the cells in half. So, at this point, we have two distinctive new cells. Each cell has the components that are necessary, including the DNA and the organelles. At this point, we have the two distinctive daughter cells. So, to wrap up, I have one more question. This is kind of an overarching question. DNA and chromosomes are extremely important to how a cell operates. I want you to explain how the process of the cell cycle and mitosis maintains the correct number of chromosomes, the correct amount of DNA, for each cell when the new daughter cells are formed. How is it that during the cell cycle, how does it account for each cell having its own DNA? What happens? Give me a brief explanation, and that's it. All right, so that does it for this. Make sure you guys finish the assignments needed and go over the study guide to prepare for the quiz. Have a great break, and I'll see you guys in the new year.